If you have chickens, you know they love corn. My flock like it best of all of the grains in their feed mix, and for good reason. It's energy dense, it's tasty, and watching chickens dash around gobbling up a handful of corn is one of the simple joys of keeping a backyard flock. But corn isn't just chicken feed. It's part of the foundation of modern agriculture. And in 1970, the United States came dangerously close to losing it. Imagine you're a corn farmer in Iowa in June of 1970. You walk out to check your fields and you notice something wrong. Small tan lesions on the leaves. Within days, those lesions spread. Within weeks, your entire field is dying. The stalks are rotting, the ears are covered in fungus, and there's nothing you can do but watch your livelihood collapse. That year, the United States lost 15% of its corn crop. That's the equivalent of about $8 billion in today's money. And the most disturbing thing? It happened because of a shortcut that seemed brilliant at the time. But to understand how we got to 1970, we need to go back much further. We need to go back 9,000 years to a river valley in Mexico where corn didn't even exist yet. In southwestern Mexico, there's a wild grass called Teosinte, and if you saw it growing, you would never ever think that this plant could become corn. Teosinte has tiny seed heads, smaller than your pinky finger, with maybe 5 to 12 hard kernels encased in a stone-like shell, like little rocks. Each kernel is individually wrapped in its own protective case, and when the seeds are ripe, they just fall off and scatter. It's not an obvious food source. The kernels are so hard, you can't really eat them. Some researchers think maybe ancient peoples chewed the stalks for their sweet juice, kind of like sugarcane. Others think maybe they discovered you could pop the kernels like popcorn to make them edible. We simply don't know exactly why anyone started paying attention to this unremarkable grass. But around 9,000 years ago, someone did. And what happened next is one of the most remarkable achievements in human history. Someone noticed a mutation, a teosinte plant where the kernels didn't have that hard protective shell. And instead of just eating those kernels, they saved them, they planted them, and the next year they did it again and again. Over thousands of years, these early farmers selected for bigger ears, more kernels per ear, kernels that stayed on the cob instead of shattering and falling off, and a plant that grew a single large ear instead of many small branches. Researchers estimate that somewhere between 800 and 1,700 genes were modified through this process of selection. Let me say that again. Ancient peoples with no understanding of genetics, no laboratories, no microscopes, somehow selectively bred for changes in up to 1,700 genes. That's not just farming. That's plant engineering on a scale that's almost incomprehensible. And it worked. By about 6,000 years ago, you could recognize this plant as corn. And by 4,000 years ago, corn had become productive enough to support the first settled agricultural communities in Mesoamerica. Corn wasn't just a crop, it was sacred. Creation stories across Mesoamerica tell of humans being made from corn. The Maya believed that the gods created people from masa, the dough made from corn. Corn wasn't separate from human life. It was the foundation of it. And as corn spread, it diversified. Corn traveled south into South America and north into what's now the United States. Different communities selected for different traits. Some wanted corn that could grow in dry conditions. Some wanted corn that matured quickly in short growing seasons. Some wanted corn for making flour, some for popping, some with different colors for ceremonies. By the time Europeans arrived in the Americas, there were thousands of varieties of corn, each one adapted to its specific place, its specific climate, its specific purpose. This diversity wasn't accidental. It was the result of thousands of years of careful observation and selection by countless farmers across 2 continents. Corn made empires possible. The Olmec, the Maya, the Aztec, the Inca, all of these civilizations were built on corn. It provided the reliable food surplus that allowed people to do things other than just survive, to build cities, to create art, to develop writing systems, to study the stars. And that diversity, that incredible genetic library built up over millennia, we're about to see what happens when we throw most of it away. Let's jump forward to the 1920s. American plant breeders made a discovery. If you take a corn plant and self-pollinate it over and over for several generations, you create what's called an inbred line. These inbred plants are weak and scraggly, not productive at all. But this is the interesting part. If you cross 2 different inbred lines together, the offspring are incredibly vigorous. They grow faster, yield more, and are much more uniform than anything that came before. This is called hybrid vigor, and you might have heard that term sort of bandied about in relation to plants in the garden and also chickens. The first commercial hybrid corn was introduced in the 1920s. By the end of the 1930s, hybrid corn made up nearly 100% of corn planted in the Corn Belt. By 1960, it had spread nationwide. Corn yields per acre more than doubled. About half of that increase came from these new hybrid varieties. It seemed like an absolute triumph, and in many ways it was. Hybrid corn could feed more people. It was more predictable. The farmers loved it. But there was a catch. Hybrid vigor only works in the first generation. If you save seeds from hybrid corn and plant them the next year, you get a mess. The genetics segregate. Some plants are good, some are terrible, and you have no way of knowing which is which until they grow. So farmers had to buy new seed every single year. Does this sound familiar? If you watched my video on the Chicken of Tomorrow contest, you're hearing a very similar story. The 1930s and 40s were the era of optimization. Make things bigger, faster, more efficient. And in both chickens and corn, we succeeded brilliantly at that specific goal. And just like with chickens, the seed companies began to consolidate. Small regional seed companies got bought up. The genetics became more and more concentrated in fewer and fewer hands. Producing hybrid seed is labor-intensive. Remember, you need to cross 2 specific inbred parent lines. That means you plant rows of both parents in the same field. And then you have to make sure only the male line parent pollinates the female line parent. Traditionally, this meant hiring hundreds of teenagers every summer to walk through the fields and physically remove the tassels from the female line plants before they could pollinate themselves. This process is called detasseling, and it's tedious and expensive. In the 1950s, plant breeders discovered something that seemed like a miracle. They found corn plants with a genetic trait that made them male sterile. They couldn't produce pollen. This trait was in the cytoplasm of the cells. Now, quick biology refresher. Cells have a nucleus, which is where most of the genetic information lives, but they also have cytoplasm, which is the gel-like substance filling the rest of the cell. And cytoplasm has its own small amount of genetic material. Plants inherit their cytoplasm only from the mother plant, not from both parents like nuclear genes. These male sterile corn plants came from a variety called Texas, so it became known as Texas male sterile cytoplasm, or T-Cytoplasm, because that's a mouthful. And it was perfect for hybrid seed production. You didn't have to pay for detasseling crews anymore. The female parent literally couldn't pollinate itself. By 1970, somewhere between 85 and 90% of all corn planted in the United States had this T-cytoplasm. There were hundreds of different hybrid varieties. Farmers could choose from many different seed companies, different brands, different names, different traits. But underneath all that apparent diversity, the cytoplasm was identical. The same T-cytoplasm in almost every corn plant in America. Farmers thought they were being careful. They bought seed from different companies. They planted different hybrids. But they didn't realize that where it mattered for this particular vulnerability, they were all planting essentially the exact same plant. And we already knew this was dangerous. In the 1840s, Ireland had been devastated by potato blight. The potato variety that Irish peasants depended on, called the lumper, had no resistance to the disease. About 1 million people died. Another 1 million emigrated. It was a catastrophe that should have taught us about the dangers of genetic uniformity. Scientists in 1970 even warned about the risk. In the spring of that year, researchers published a paper noting that although there were many different inbred lines being used, giving genetic variability among hybrids, the cytoplasm of the American corn crop was essentially uniform. They said, and I'm going to read this here, that corn was uniformly susceptible and exposed to a pathogen capable of developing in epidemic proportions. That paper was published in the spring. By summer, their worst fears were realized. In 1969, a few farmers in the southern United States noticed something strange. A disease called Southern Corn Leaf Blight, which had always been a minor and manageable thing that happened to corn, was suddenly much more aggressive. By 1970, it became clear why. A new race of the fungus had emerged. Scientists called it race T, and race T had a particular talent. It could produce a toxin that was absolutely devastating to corn with T-cytoplasm. The toxin didn't just damage leaves, it infected stalks, ears, and developing kernels. The disease spread north through the summer of 1970 like wildfire. By August, it had engulfed most of the southern corn belt. By mid-August, it had spread as far as Nebraska, Minnesota, and all of the eastern United States. Corn futures on the Chicago Board of Trade jumped from $1.35 per bushel in late July to $1.68 by mid-September. The Wall Street Journal and the New York Times ran stories about the epidemic. There was genuine concern about whether the United States could feed its livestock through the winter. In the end, the country lost about 15% of its corn crop. In today's dollars, that's roughly $8 billion. Some states lost much more. In some southern states, losses reached 50%. The response was swift. Seed companies immediately switched back to using normal cytoplasm. They hired those detasseling crews again for the 1971 season. And remarkably, that's all it took. The crisis was over. Yields returned to normal. Race T still exists, but it can't infect corn without T-Cytoplasm. So, we learned our lesson, right? We understood the dangers of genetic uniformity, and we made sure to maintain diversity in our crops. Well, not exactly. Today, just 4 companies control more than 60% of global seed sales. The same companies that make the chemicals sell the seeds that are bred to work with those chemicals. The seed industry is more consolidated now than it was in 1970. And we're facing this reality at exactly the moment when we need genetic diversity the most. Climate change means we can't predict what conditions we'll need crops to tolerate. Drought, floods, heat, new pests, new diseases. The genetic solutions to those problems might exist somewhere in the thousands of varieties of corn that were developed over millennia. A period of time that went through multiple different climatic shifts. Or they might not, if we've let them go extinct. I'm growing 2 heritage varieties of corn in my garden right now, a heritage sweet corn and a heritage flint corn, and I'm doing it for the same reason I keep heritage breed chickens. Not because I think these varieties are going to replace our modern agriculture, but because I believe in preserving our options. We can't know which genetics we're going to need in the future. Once a variety is extinct, all those genes, all those traits that were selected for over generations, all that adaptation to specific places and conditions, it's gone, and we can't get it back. This story of corn is a story that keeps repeating itself, with chickens, with potatoes, with wheat, with almost every major crop we grow. Optimize for one trait, concentrate the genetics in fewer hands, and suddenly you're vulnerable in ways you didn't predict. In 1970, Race T caught us by surprise. We recovered quickly that time, but next time we might not be so lucky.